Uh, Hi everyone. Katya Vikita is associate professor at the Faculty of Science of the University of Lisbon. Her research explores how ontologies, knowledge graphs, and artificial intelligence can work together to transform heterogeneous data into explainable scientific knowledge. She has made significant contributions to semantic data integration, ontology, and knowledge graph matching, biomedical knowledge representation, and explainable AI. Her work is strongly aligned with the goal of governing cost action, particularly in advancing the construction, integration, quality, and reuse of cross-domain knowledge graphs. She has been invited international keynote speaker and has collaborated in several national and international projects with academic, healthcare, and industrial partners. She is also committed to promoting computer and data science as a fundamental instrument for scientific research and encouraging young women to pursue careers in these details. Thank you, Katia. Now you have the floor.
Thank you so much for having me, and thank you for the invitation, Eleni, and for you. It's really a pleasure to be here. So before we were officially starting this presentation, I was sharing that it's been a while since I've been able to talk about the work we've been doing in my lab on ontology and knowledge graph alignment and integration. So I'm very, very happy to have this opportunity to share with you some of our, I would say, most interesting work in this area and how we are using this ability to integrate knowledge across domains to support explainable artificial intelligence in scientific discovery. So the first thing that I wanted to share with the audience is why in my lab we believe that scientific discovery needs knowledge graphs. And we actually have experience over the last nearly 2 decades already that knowledge graphs are actually key to solve or to address some of the most fundamental problems in using machine learning and artificial intelligence for scientific discovery. So the first challenge is that there are immense amounts of data that are being shared in non-standard formats. And I don't think that I need to explain to this audience that the challenges of converting, interpreting, merging all of these data into readable formats can be really made a lot more easy if we have the standards, if we have ontologies that cover these domains, and then if we can articulate this heterogeneous data in a knowledge graph. The other interesting challenge is that data that subsumes scientific discovery is heterogeneous and complex. And many of the current machine learning approaches are processing these very large data sets without connecting the underlying data to context, to meaning, to scientific knowledge that we have accrued over decades or maybe even more than a century. So knowledge graphs also can play a fundamental role in connecting data to the scientific context, to the scientific knowledge that allows machine learning to interpret data in terms of scientific validity and plausibility. But on the other side, there are also areas where we want to be able to apply machine learning approaches such as deep learning, but we can't because there's a lack of sufficiently large data sets. Areas such as rare diseases or areas where experiments are too expensive or too dangerous to conduct. And so there's also here an opportunity for knowledge graphs to come in and be used to actually inject a layer of knowledge over this data that allows machine learning to work over fewer data by either generating synthetic data based on the knowledge graphs or by allowing less complex machine learning models to work over fewer data, but achieve better performance by having this layer of contextual knowledge to explore. And finally, and what is mostly the topic of today's talk, explainability. So There's no science without explanation. Science is the process by which humans try to understand the world, the universe, existence. So without explainability, we can not have science. And if we want to be able to use artificial intelligence methods and machine learning methods to support scientific discovery, then these need to have an explainability layer, a way to communicate to human scientists what are the discoveries, whether they fit or contradict existing knowledge, existing scientific knowledge. And again, by allowing us to have this shared substrate of context and meaning, ontologies and knowledge graphs can be key to tackle all of these challenges. So today I want to share with you a case study in biomedical research that explores something that I find super interesting. So I think that many of the most interesting questions in research nowadays cross-cut domains. It's impossible to answer them by just looking at data from one type in one domain. It's impossible to answer these questions by being siloed. So the question that we ask ourselves, so how can we support the application of machine learning methods and explainable AI methods when the underlying data is not only complex, as is the case in biomedical research, but is also heterogeneous, cross-cuts domains, and is potentially incomplete and even ambiguous. So for this, one ontology or one knowledge graph that only covers one domain is definitely not enough. In areas such as systems biology or systems medicine or personalized medicine, what we require are holistic representations. So representations that cover the whole gamut, down from the molecular level where we can be, looking at protein functions, small biomolecules, drugs, but also to the large scale of clinical events and environmental factors, and even how we conduct research about these topics. And to be able to cover, molecular, cellular, anatomy, phenotype, disease, clinical, environmental aspects, we need to be able to integrate knowledge across both the scope and the granularity. So let me give you an example of a recent European project that we worked on. And the goal of this project was to produce predictions of what is the best drug for a given cancer patient by looking at enormous amounts of data about that patient, including mutations and transcription, so omics data. And we wanted to be able to not just recommend a drug, but to explain both to clinicians and researchers why that drug makes sense for that patient. The problem here is that this is a multi-domain problem. So if we just pick up existing ontologies and try to map out the data that we had for this project, we would end up with a disconnected graph. We would have some information about genes and drugs on one side. We would have some information about the mutations of the patient and other features of the patient. We would have some information about the high-level topics on the area of oncology, but by just having these independent ontologies, we would miss important links. So we started out by this idea. So actually, this diagram that you're going to see right now was designed before any algorithm or any strategy was developed. So we started by designing our goal, our wish list for an end state. So we wanted to be able to find mappings across all of these ontologies that would break down this domain silos and would allow us to connect everything from patient-relevant features down to the drug recommendation, and here the drug is sunitinib in yellow, and to do so in a way that would produce explanations that would allow researchers to understand whether that recommendation actually makes sense from a biological point of view. And you see here actually that the explanation is a path in the graph. Okay, so we can understand here that if our John Doe patient has a mutation in the MET gene, in the position T54, in the position 540 that changes a T for a G in that gene, that is going to promote the transcription of that gene, which is related to tyrosine kinase activity, that's an enzyme activity, which is actually inhibited by the drug. So a patient with a mutation has more activity of that protein, but the drug inhibits that protein. And this shows why that drug makes sense for that patient, because he has that mutation whose downstream effect is inhibited by the drug. So this was our starting point. How can we get to a graph that is integrated at this level of richness? So the generic question is, what do we need to create holistic representations? And so on one hand, we want to be able to cover multiple domains. We want to be able to align multiple ontologies, from cellular, molecular aspects, clinical aspects. And since we're talking about multiple ontologies, one concern is, of course, scalability. If we're talking about aligning tens of ontologies, how does that work? And the other challenge is to ensure that rich and deep semantic integration. We have a question here because related but not equal domains. So the domains are related, but they're not equal. It's not just a matter of finding an equivalence link between the ontologies. We need to find related links, but also maybe even more complex relations that involve more than one ontology, if we want to produce this overarching holistic representation to drive scientific discovery and to drive explainability. So let's cover some basics. So just to remind you a little bit, the classic definition of ontology matching is to find a function where we have as input a pair of ontologies and potentially a pre-computed alignment that we want to extend, a set of parameters and resources, and that's going to return an alignment between these ontologies that is composed by all of the mappings between the entities in those ontologies. And these mappings are typically equivalence mappings between classes and properties of an ontology. And that's kind of the standard state-of-the-art for ontology matching. But we want to do a little bit more than this, right? Because we want to create those holistic representations. So we not only want to do this for many, many, many ontologies, but we also want to do this across multiple ontologies. So we want to find mappings that are complex mappings, where at least one of the mapped entities is an expression. So for instance, in this very simple example, what is one class in one ontology, accepted thesis proposal, is equivalent to the intersection in the second ontology of the class thesis proposal, but also the property accepted by. And if we transpose this into I'm sorry for this. And if we transpose this into a biomedical example, we have even more complexity, because now instead of just having two ontologies that we want to link with complex mappings, we can actually have complex mappings across more than two ontologies. So for instance, here, a mapping for the human phenotype concept decreased circulating cortisol level is actually a very complex OWL expression where we use both properties and classes from a number of different ontologies. But when we wanted to design that integrated knowledge graph that supports explainability for drug recommendation in cancer, we were first looking, okay, what is already out there that can be reused for this? So on one hand, we have ontology alignment systems that find and they're able to find a set of mappings between entities in two ontologies, most of them equivalence mappings. We have mappings repository for biomedical ontologies that we could potentially reuse. And there's also logical definitions that are defined within biomedical ontologies. And these are these complex definitions of a class in one ontology by composing classes of other ontologies. So a little bit like a complex mapping. So here, hearing impairment is equivalent to having a part that inheres in some decreased rate of sensory perception of sound that is abnormal, and that gives you hearing impairment. But the problem is that, first, available mappings are simply not enough. So right now, the bioportal repository has over 1,200 ontologies and 64 million mappings. But these mappings are typically close match, related match. They've been made using naive string matching or recomputing over existing taxonomies from biomedical thesauri, and they do not afford that richness of semantics that we wanted. Moreover, the open biomedical ontologies logical definitions, which do afford that semantic richness, are very few. Okay, the number of ontologies that have this definition is small, and the percentage of coverage for those logical definitions, with very few exceptions, is also very, very, very small. And in a third challenge, the state-of-the-art ontology mapping systems at the time when we started working on this, so this was 2020, 2021, was also not enough. So here I have updated statistics to show you that it is actually still not enough today, because the state-of-the-art in symbol equivalence, although it achieves very good results in some areas, such as anatomy with 94.1 in F1 score, actually struggles to achieve results that can support this high quality integration of biomedical ontologies, because we have F1 scores between 70s and mid 80s in all of the other tracks at the Ontology Alignment Evaluation Initiative tracks that use biomedical ontologies. So this kind of tells us that, okay, reusing mappings is not enough, logical definitions don't have sufficient covering, and using state-of-the-art systems is also not going to really be enough. So this led us to think that we need to kind of rethink biomedical ontology alignment. So on one hand, we want to cover multiple domains, and we want to move from this paradigm of just pairwise ontology alignment to the holistic alignment of multiple ontologies. But we also want to ensure a rich semantic integration. And so we want to move from simple equivalence ontology alignment to complex ontology alignment. So the first challenge is the holistic ontology alignment. So when we want to align a lot of ontologies in the biomedical field, that quickly becomes intractable. You have here a subset of all of the ontologies that we wanted to align to build that knowledge graph. And as you can see, the vast majority of them contains more than 10,000 classes. So matching all combinations, all pairwise combinations of all ontologies, when these ontologies are extremely large, quickly runs into an intractable problem. So we find that we have both a scalability issue, we have an issue of guaranteeing a coverage of all subdomains, and we also want to guarantee high quality mappings. So classical strategies for holistic ontology matching, which match multiple ontologies into a single alignment using equivalence mappings, apply one of two strategies. The first one is partitioning the search space. So we just come up with pairs of ontologies, merge those pairs to create new ontologies, and then merge the merged ontologies until we arrive at a final integrated ontology. Or incremental matching, where we just take a pair, match, then match a third ontology to the already matched pair, and so on and so forth. But both of these strategies have questions. So in the first one, where we are partitioning the search space, the first question is, how should we partition the search space? Randomly, non-randomly, what will guarantee us not only the scalability, but the coverage and the high quality? And in parallel, in the incremental matching approach, then the question becomes how to order. Order will have an influence in how well you're going to cover some subdomains and how good your end alignment is going to be. So we actually decided that a good strategy would be to combine both paradigms. So for this, we were reusing agreement maker lights matching algorithms, which is this tool for ontology matching that we've developed in the lab for many, many years, and it performs really well and really fast for biomedical ontology alignment. And we developed first a partitioning approach, and we decided that we would partition based on semantic overlap, and I will explain in a bit how that works. And then we also complemented this with an incremental matching approach that also used the semantic overlap to define the order in which we would combine these ontologies. And then we extended AML to be able to not just match the ontologies, but also merge ontologies and import the ontologies. So how does this all start? So it starts with what we call an anchoring. And the anchoring is going to define what is the semantic and lexical overlap for all ontology pairs by considering first what's already represented in the ontologies. So biomedical ontologies have a lot of cross-references that we can explore. They also come with logical definitions that we explore to compute this semantic overlap, and then also reused classes and properties and lexical similarity. And based on this overlap, we can produce a semantic affinity matrix between all of our ontologies. And this is going to be used to both to partition the space through clustering and to define the order for the incremental matching approach within each cluster. And so we will define this order by first matching the ontologies that are most similar to each other. So when this was done, we actually had this experimental design where we wanted to compare a global pairwise approach, the standard approach. Let's do all of the pairwise matchings, all versus all of those ontologies, which is actually a 378 tasks in ontology alignment. And then compare that to our holistic matching, where we are both going to test both clustering pairwise and clustering incremental based on this semantic anchoring, based on the semantic overlap. And what our results found was that, especially for our incremental clustering, the total time to run an alignment was cut down by half without sacrificing quality. So for our global pairwise, just doing all versus all, we took around 30 hours. But then when using the incremental clustering, we cut this down for a little over 15 hours. But this is not the end of the story, right? Because we also want to go beyond simple equivalence matching. So we now have sorted simple equivalence matching in terms of scalability. We have a baseline of equivalence mappings for holistic matching, but we need to go beyond simple equivalence matching. And here, I want to just give you first a landscape of what we mean by beyond simple equivalence matching. So there's subsumption matching, where you have two ontologies with a similar domain and different granularity, and you're mostly establishing subsumption mappings. Then you can also have standard complex matching, where you have two ontologies. They cover a similar domain, but with a different model. So you may need to combine classes and properties of one ontology to match to another. But then what is more interesting to us in the field of biomedical discovery is when you have multiple ontologies of different domains, but you need to connect them to make sense of a very complex problem. And that brings us to complex multi-ontology matching. So in this case, we would need to use 3 different ontologies, orange, blue, and green, to produce an equivalent class to abnormal immune system cell morphology. So let me show you a real mapping of something that we want to be able to produce. It is quite complex because in this mapping, to map to decreased circulating cortisol level in the human phenotype ontology, we need to do 2 things. So we need to find all of the classes and properties to compose and find a structure, the logical construct in OWL on how to combine all of this to produce an expression that is equivalent to the original class. And that brings so many challenges. The first one is that there are no theoretical restrictions on the arity of the target expression. So this means that the target expression can include any number of distinct entities from any number of ontologies. And that increases the complexity of these strategies a lot. But then, to make things worse, they can also be combined in any manner conceivable within the expressivity of our language to produce the logical construct, right? The complex expression. So you can see why this is really, really, really complex, right? So we broke down this problem in actually two challenges. So the first one is dealing with what's in blue. So how can we deal with finding out any number of distinct entities out of any number of ontologies? And that's like candidate class generation to be composed in a complex expression. And then we have the second problem is when we know the ingredients, how can we find the recipe on how to combine them to produce this complex expression? So let's focus first on candidate generation. So our intuition here was that the anchor, so the thing that connects, the connective tissue in biomedical ontologies is actually the lexical component. And this means the labels, the synonyms, all of the textual component of the ontology is where a lot of the relevant information to extract these complex mappings exists. So we actually did a two-pronged approach. A very simple one, which is just based on a lexical approach and overlaps of labels, and a more complex one, which is actually based on language embeddings. And then by combining the results of those two strategies, aggregating, filtering, we produce a final set of class candidates. But let's see this in a little bit more detail. So let's start with our lexical class combination. So we have the source name, a string, and we want to find a combination of target names that affords maximum coverage of the source name. And so we do this by combining target names from all of the potential ontologies in any combination possible. So this is quite expensive to run. But we also complement this with something that gives us a lot more flexibility in terms of lexical matchings, because this is constrained by how exactly you write down your terms, but we want to have something that actually focuses on the meaning of these terms. And for that, we used language models, in our case, sentence bird embeddings. And it's actually based on a very simple idea. So we just convert all names in the ontology to embeddings. And then we take the embedding of the source class that we want to map and find what is the most similar embedding in all of the ontologies that we have at our disposal. And these can be 10 ontologies, 20 ontologies, very many ontologies, and we find the most similar one. So we start here with the embedding for our source, and we find the most similar one. In this case, our source is shortening of the Taylor neck, and the most similar one is Taylor neck. And then we do a smart thing. Then we just compute a geometric operation over the embedding that removes Taylor neck from shortening of the Taylor neck. And that generates a synthetic embedding here that represents the meaning that is left on the source when you remove the first target candidate. And then you ask again, what is the most similar embedding to this one? And in this case, it was decreased length. And decreased length actually matches shortening, right? So it's a deceptively simple approach that allows you to compose the meaning of the original class by computing these geometric operations over the language model embeddings for the ontology classes. So how good were we at generating candidates? So we actually have 3 partial references that were constructed from existing logical definitions. If you remember from the beginning of our talk, logical definitions are a type of complex mapping. And then for these partial references, we computed semantic precision, recall, and F1 score, and we compared our strategy, CMOM-RS, with two ablated versions, so without the language model and without the lexical component. And what we found is that the lexical component, so when we remove the language model, is actually more precise. We get better semantic precision. And the language model, of course, provides more coverage, so increases recall, which is actually the behavior that we were expecting it to do, right? Because it allows us to move beyond the strict lexical overlap. And so in 2 out of 3 cases, we were able to achieve the best F1 score with our candidate generation approach. But we still have more complexity to solve because candidate generation produces only sets of candidates and not the full construct. We still want to compose all of those ingredients in the correct complex expression that matches our source class. So the question was, how can we teach language models to reconstruct the patterns that we need? And so the idea is that we combine our candidate generation with extracting some complex mapping examples that we can use in an in-context learning approach to let the language model, by looking at our selected class candidates and examples on how to compose them into complex expressions, produce novel, complex multi-ontology mappings. So what we actually want is to build an instruction set for in-context learning that shows our language model not just relevant patterns on how to compose complex mappings, but also give us input, the class candidates. Because if you remember, it's finding that those class candidates that really expands your search space, because there's no limit on the arity, right? The number of entities and the number of ontologies where they can come from. So first, what we do is reduce the search space by using that smart candidate generation approach, and then just prompt a language model to, by looking at cleverly selected patterns, and we will get to those in a minute, and the classes that you selected.
Yeah, sorry.
Yes.
We're approaching time, so if you could just wrapping up.
Okay.
Thank you so much. Sorry to interrupt.
That's okay. To produce this final mapping. So we extract these examples by using a named space abstraction and cardinality preservation. So we show patterns that can be composed with those candidate classes. And we use these patterns by using those reference samples. And then we compose a set of instructions for complex mapping, and we evaluate them by comparing a logical definition to what we produced. Since it's not easy, because it's not just a case of matching one class or one property, we developed a graph edit distance that allows us to compare piece by piece how well we did in reconstructing the complex mapping. And our results show that our approach, CMOM-Gen, actually achieves better precision, recall, and F1 score across all baselines, both our original candidate generation with and without examples. And so having this in-context learning approach with examples and candidates is better in most experiments. And we also asked experts to rate novel mappings because we only have partial reference mappings. So to rate novel mappings in terms of their quality, and 60% of novel mappings were rated as high or very high quality. So just to finish, what we wanted was to produce this knowledge graph that has all of these richness. And that knowledge graph is ECKO, Explainable Clinical Knowledge for Oncology. And it goes beyond existing knowledge graphs for biomedical discovery because it includes these types of mapping. So existing knowledge graphs such as PrimeKG and others only integrate by having a shared IRI, But we built ECKO by using both holistic ontology matching and complex ontology matching. We mapped 33 ontologies, 74 datasets, 1.4 million ontology classes, 112 million individuals, and 330,000 mappings. And this allowed us to produce those explanations. And you can ask me in the questions if you want to learn about how to produce these explanations. I'm sorry about this. I thought I had 40 minutes, but actually it was 30. So let me move along. And these explanations look like paths in the graph that traverse some of those complex mappings. And actually, the majority of explanations that we were able to generate actually traverse these mappings, something that a simple ontology and KG integration would not support. And mapping-based explanations are equally informative, but considerably shorter. And shorter explanations boost interpretability. So my take-home message is that knowledge graphs are a key component for explainable AI in scientific discovery, and that explaining hypotheses that cut across domains requires deep semantic integration of knowledge graphs. Of course, none of this work would have been possible without amazing past and present students and collaborators, and a picture of my group. And just one final thing, I would like to dedicate this talk to the person who got me hooked into ontology matching and knowledge graph alignment, Professor Isabel Cruz. Thank you.